a molecular dynamics study of non-covalent interactions between Si-Ppcs and biomolecules. I'm Frederico Henrique Ferreira, and I work alongside Professor Luiz Antônio Costa. Cancer chemotherapy is widespread on Earth. Cisplatin was the first drug that led to many analogs. BBR-3464 was the precursor of polynuclear platinum complexes, introducing a new mechanism of action. Si-Ppcs don't have substitutions once inside the cells, and acts only non-covalently. Mono, di, tri, and tetraplatinum is in the scope of our work. These complexes interact with biomolecules such as DNA and HS, which is a linear polysaccharide responsible to many biological functions such as vascularization, angiogenesis, and cellular recognition. Those non-covalent interactions are named sulfate clamps and forks, stabilized by a ring of 6 and 8 atoms respectively. PPC's platinum centers are highly selective for oxygens. We intend to foresee those interactions and evaluate it in terms of both energy and structure changes in order to find possible keys to help cancer treatment. We've performed a 200 nanoseconds molecular dynamic simulation of our Si-Ppcs HS systems as shown, and figure out important information concerning our MSD, our MSF, interaction energy, 8 bonds, and surface area accessible to solvent. The temperatures increased in intervals of 50 Kelvin during heating steps. Also, other physical properties assure the stability along the trajectory. Our MSD concluded a significant decrease in structural deviation of HS based on the presence of the complexes. Both diplatin-NC and A844 showed as the most rigid, whereas tetraplatin was the least. Our MSF indicated that atoms in the center of HS decreased their fluctuation in the presence of certain platinum complexes. Monoplatin-NC, A878B, and diplatin-NC as examples. All behaviors can be seen in this graph. The interaction energy analysis was able to show there is no preferable sulfates in the biomolecule, and the mean values indicate both tetraplatin and triplatin-NC as the most stable ligands, followed closely by A878B. Generally, the higher the number of platinum centers, the lower the energy. Also, solvent-accessible surface area reveals a decrease in the area that solvent can access. At last but not least, H-bond showed that the studied interactions occur in about 20% or even more of the observable hydrogen bonds. To sum up with, HS interactions are similar to those with DNA. Although more carbon atoms didn't affect the energy, the number of metallic center affected a lower Gibbs tree energy. The interactions occur mainly with sulfate groups in the middle of heparan sulfate, with either a clamp or a fork structure. We would like to thank CAPS CNPq, FAPEMIG, Federal University of Juiz de Fora, and NEQC for all resources provided. Thank you very much indeed.